Let's review a couple key concepts from this unit to prepare for benchmark 4, starting with DNA structure. DNA is shaped like a double helix or twisted ladder. Here's a picture of the double helix. We can see that the double helix is made firstly of the sugar phosphate backbone, shown here in yellow. This makes up the outside of the DNA molecule, or the sides of the ladder. The nitrogenous bases make up the rungs of the ladder, or the inside of the molecule. These are adenine, thymine, cytosine, and guanine. The nitrogenous bases follow specific pairing rules. Adenine pairs with thymine, and guanine pairs with cytosine. You can remember apple on the tree and car in the garage. A pairs with T, C pairs with G. Let's look at the structure of a single nucleotide. Nucleotides are the building block of the DNA molecule. We can see that a nucleotide has 3 parts: a phosphate group, shown in yellow, a sugar, shown in blue, and a nitrogenous base, shown here in green. The nitrogenous base shown in this picture is adenine. Thymine, cytosine, or guanine could also appear in this position. Here's a more detailed picture of a DNA molecule that shows how the nucleotides fit together. We can see a more detailed picture of the sugar phosphate backbone, and we see that in that backbone, there's alternating between the sugars and the phosphates. So it goes sugar, phosphate, and so on. The sugars here are shown in purple, and the phosphates are shown in yellow. We can see that they alternate in the sugar phosphate backbone. We see the nitrogenous bases making up the inside of the molecule, and we can see that the pairing rules are being followed. A pairs with T, and C pairs with G. Apple on the tree, car in the garage. Let's recall the purpose of DNA. This picture shows what's called the central dogma of molecular biology, which states that DNA is transcribed into mRNA, which is then translated into protein. So the point of DNA is to tell the cell what proteins it should make. Proteins determine traits and perform several essential jobs in the cell. RNA is the intermediate step between DNA and protein. Its structure is similar to DNA, but with a few key differences. Firstly, RNA is single-stranded. So we can see in this picture that RNA is a single helix, not a double. Furthermore, RNA has a different backbone structure than DNA. It has a different sugar that breaks down much more quickly. Lastly, RNA contains the base uracil instead of the base thymine. So in RNA, instead of A pairing with T, A pairs with U. So you can remember apple under. In summary, here are the differences between DNA and RNA. DNA is double-stranded, whereas RNA is single-stranded. DNA contains thymine, whereas RNA contains uracil. And lastly, DNA contains a deoxyribose sugar, whereas RNA contains ribose. DNA replication is also going to be on the benchmark. This is the process by which DNA copies itself. The point of doing this is to double the amount of DNA so that when the cell divides, each daughter cell has the correct amount of DNA in its nucleus. The process is semi-conservative. That means that each new DNA strand contains one old DNA strand and one new one. Here's a video on the replication process. I'm going to include a link to this video in the Edpuzzle. Go ahead and follow the link, pause this video, and return once you've watched the video on semi-conservative replication. So pause, navigate to the link, watch the video. Let's now move on. As we saw in the video, when DNA copies itself, each new DNA molecule contains one old strand and one new one. Here's a visual of this process. We can see that each new DNA molecule has one old strand, shown in blue, and one new strand, shown in red. So again, that is called semi-conservative replication. The last things to know about DNA replication. You should know that DNA replication is carried out by enzymes. The first one to know is helicase. Helicase breaks the hydrogen bonds between nitrogenous bases and splits the double helix. Here we can see the helicase, shown in green, splitting the DNA molecule. You could think of helicase as unzipping the DNA. The other enzyme to know is DNA polymerase. DNA polymerase takes the unzipped DNA and adds new nucleotides to it. This allows the DNA to copy itself. So DNA polymerase does the actual copying by adding new nucleotides to each exposed DNA strand. At this point, I'm going to have you follow another link and refresh your memory on how to transcribe and translate a DNA sequence. Once you've spent 5 or 10 minutes practicing, return to this video. So follow the link, practice transcribing and translating, and then return to this video when you're done. Now that you've brushed up on your transcribing and translating, let's talk about what transcription and translation are. This will be our last topic for today. So, transcription takes DNA and turns it into mRNA. Translation takes that mRNA and turns it into a protein. Here's an image of transcription taking place. The kidney bean-shaped protein in the middle of the screen is called RNA polymerase. We can see that the RNA polymerase is reading the DNA molecule. RNA polymerase is then going to produce a strand of mRNA from that DNA molecule. So the point of transcription is to take the DNA and use it to make mRNA, which is what we're seeing going on here. Once transcription is done and the mRNA is made, the mRNA will exit the nucleus and then be translated into a protein. Here's a picture of translation. During translation, the mRNA molecule gets red. So the mRNA strand here is shown in red, and the turquoise protein is called a ribosome. The ribosome is reading the mRNA molecule. We can see that the mRNA is read in groups of 3. Those groups of 3 letters are called codons. The tRNA molecules, shown in yellow, have anticodons on them. The anticodons and codons match with each other. This allows the tRNA to bring the correct amino acid to the ribosome. We can see that those amino acids are being joined together in a specific order to make a protein. So in summary, the mRNA is read in groups of 3 letters called codons, and then the tRNA uses its anticodon to bring the correct amino acid to the ribosome, which is then added to the growing protein strand.